Welcome to our webinar about the effect of different SARS-CoV-2 variants on COVID-19 assays and vaccines, organized by Antibodies Online in cooperation with Nanotemple Technologies. My name is Stefan Pellens, Product Manager at Antibodies Online, and I was supposed to be presenting today with Andreas Langer, Senior R&D Scientist at Nanotemple. However, because of a non-COVID-19 related medical emergency, Andreas could unfortunately not join me today, so I am covering his part as well to the best of my knowledge. This installment is an update of a webinar presented back in September last year, so before the emergence of the SARS-CoV-2 B.1.1.529 Omicron variant. For those listeners that have already attended the previous presentation, I would like to note that the provided background information has remained largely the same, with the addition of new findings of the last few months. I will also be showing additional data that we have produced over the course of the winter, most notably pertaining to the Omicron variant. The webinar will be recorded, and we will make the recording available on our website within the next week. Any personal information about participants will be removed, so please don't hesitate to use the chat window to ask any questions. These questions will then be answered in the Q&A session subsequently to the presentation. Let me give you a short overview of today's presentation. So first, after a few introductory words about today's organizers and speakers, I'm going to talk a little bit about the SARS-CoV-2 proteins, and in particular the S protein and its significance in context with existing vaccines. Subsequently, I will take a closer look at the S protein structure and the effects of some of the mutations in various virus variants, and I will provide some background on one exemplary anti-S protein antibody. I will then present detailed affinity data that we have produced for this antibody in context with the S proteins from different SARS-CoV-2 variants, as well as for some other antibodies. After putting this data in perspective to some real-world data, we conclude the presentation with a quick teaser about some ongoing experiments and some summarizing remarks, followed by the Q&A session that I had mentioned before. Organizers of today's webinar, Antibodies Online and Nanotemple Technologies. Antibodies Online was founded in 2006 as a spin-off of the Aachen Technical University. We are now located still in Aachen in Germany and also in Philadelphia, Pennsylvania. Our mission is to make the search and purchase of antibodies, proteins, kits, and other proteomics products easy and straightforward. We count more than 6,000 institutions in 65 countries as our customers, some of which are indicated here. In April 2020, within 1 month after COVID-19 had been declared a global pandemic, Antibodies Online offered about 600 SARS-CoV-2 related antibodies, proteins, and kits. By May 2020, so 1 month later, many of these had already been delivered to customers in over 25 countries. And by now, so as of March 2022, we have delivered an ever-increasing number of proteins to more than 500 labs worldwide. This confirms Antibodies Online as a trusted source for COVID-19 research. Besides great product, we also strive to provide great content relating to these to the research fields that these products are being used in. We have, for example, released the world's first product-centric SARS-CoV-2 mutations tracker to help our customers keep pace with the highly dynamic COVID-19 research field. We have also compiled a SARS-CoV-2 handbook, providing an overview of the SARS-CoV-2 virus, COVID-19 biology, and related products. The mutations tracker and the handbook are freely accessible on our Antibodies Online website, and we did actually just put the latest iteration of the handbook out online today, so please go ahead and download it. Validation data plays a key role for us at Antibodies Online regarding our products. We pick and recommend products that come with the best data. And we do also generate additional data through our independent validation program. And the data that I will be presenting today is one of the results of this endeavor. Nanotemple Technologies was also founded as a spin-off of university and in 2008 of Ludwig Maximilians University in Munich. The company is headquartered in Munich in Germany, and it has by now 8 additional offices in Europe, the US, China, India, and Japan, with a total of approximately 160 employees. The product portfolio comprises 4 different product lines for the characterization of proteins. These are the monolith and dianthus platforms, which use the effects of microscale thermophoresis and temperature-related intensity change to measure molecular interactions. And on the other hand, there are the Prometheus and Tycho platforms, which apply nanodifferential scanning fluorometry to measure protein stability. All in all, more than 1,000 Nanotemple instruments are used around the world today and were cited by now in more than 5,000 publications. As speakers for today were foreseen Dr. Andreas Lange and me, Stefan Pellens. But while Andreas can unfortunately not present today, let me mention that he is the one mainly responsible for the experiments of which I will be presenting the data today. Andreas holds a doctorate in biophysics from the Technical University of Munich and has extensive experience in protein interactions. As an application scientist, Andreas has advised Nanotemple Technologies distributors in Japan, India, and South America on the use of the different Nanotemple instruments and platforms. He is now Senior R&D Scientist at Nanotemple Technologies. Me, on the other hand, I have obtained a PhD in genetics from Université Paris 7, followed by a postdoc at the University of Washington in Seattle, Washington. Both during my PhD and postdoc work, I have worked with engineered DNA endonucleases, and I have therefore a long-standing interest in protein structure and how it influences protein function. I am now working as a product manager at Antibodies Online, and I am responsible also for our independent validation program. So, which brings us finally to our topic of today's presentation, the SARS-CoV-2 virus and its proteins. SARS-CoV-2, the causative agent of COVID-19, has a positive sense single-stranded RNA genome with a size of almost 30 kilobases. This genome encodes a total of 26 proteins. 16 of these proteins are so-called non-structural proteins. That means they do not contribute to the structure of the assembled virion. Their functions are primarily downregulation of host protein production, virus replication, and production of new virions once SARS-CoV-2 enters a host cell. Among these are the host shut-off factor, NSP1, the two virus proteases, PLP-Pro and 3CL-Pro, or the M-Pro for the main protease, and the RNA-independent polymerase and exonuclease for proofreading during genome replication, which is the main reason for the comparatively low mutation frequency of the SARS-CoV-2 genome. In addition to the SARS-CoV-2 genome, the SARS-CoV-2 genome contains a sequence for 9 accessory factors whose function in virus replication, modulation of host immune response, and packaging remains largely unclear. And lastly, there are 4 structural proteins encoded in the genome, the spike, envelope, membrane, and nucleocapsid protein that make up finally the assembled virion. For this presentation today, I will focus mainly on the, solely actually, on the spike protein marked here in orange. So why the focus on the spike protein? To illustrate this, I list here some SARS-CoV-2 vaccines that have been approved for use in humans. They rely on different technologies. The well-known BioNTech and Moderna vaccines built on RNA encoded by encoding a viral protein, which is then packaged in lipid nanoparticles for delivery. Other vaccines, like those from AstraZeneca and the Sputnik V vaccines and Johnson & Johnson's and the CanSino Biologics vaccine, rely on delivery of viral S protein DNA and subsequent production of the antigen by the infected cells. While the delivery and the genetic information for the antigen varies between these vaccines, they are all solely relying on the S protein to mount an immune response against SARS-CoV-2. The Novavax and the SARS-CoV-2 vaccines are based on a virus protein, and which is a first vaccine, circular DNA, respectively. But again, in spite of the different platforms, the S protein is also here the pertinent antigen for both of these vaccines. The two vaccines that are shown here on the bottom row of this table, the Sinovac vaccine and the Indian Covaxin, use inactivated SARS-CoV-2 virions. Therefore, they display also the other structural proteins as opposed to only the S protein. However, the S protein appears to be the most immunogenic of the SARS-CoV-2 structural proteins, and one can assume that even in these whole virion vaccines, the S protein plays a crucial role to elicit an immune response. Besides, the S protein is also the host factor's point of contact with the S2 receptor for virus entry. So in short, the S protein of SARS-CoV-2 is very important for all COVID-19 vaccines, independent of selected platform. And in addition, it's also the most relevant target for antibody-based SARS-CoV-2 immunoassays. So let's have a closer look at the SARS-CoV-2 S protein. In order to better understand the body's interaction with SARS-CoV-2, it is helpful to have a better understanding of the S protein structure. The S protein protrudes from the spherical virion, shown here on the left, and gives it its characteristic appearance, resembling a crown that has earned the group of coronaviruses its name. Zooming in on the S protein here on the right shows that it's actually a homotrimeric complex consisting of 3 monomers, shown here in yellow, in gray, and in green. The lower S2 section of the complex contains a trimerization interface and the membrane proximal stalk that is important for membrane fusion and virus entry. The upper S1 section contains the N-terminal domain, or NTD, and the receptor binding domain, or RBD. The 3 RBDs for the yellow, gray, and green subunit shown here have to transition from a closed into an open position, shown here on the right, in order for the virus to make successful contact with the S2 receptor on the host cell surface. While the NTD has been shown to contain many immunodominant regions eliciting a T cell response, the RBD is thought to be the primary target for antibodies. Therefore, mutations within this RBD, which is shown here now in orange, are highly relevant for the effectiveness of antibodies, either in the host's immune response or in immunoassays. Having a closer look at the S protein mutations present in SARS-CoV-2 variants of concern and some of the variants of interest reveals several amino acid changes that can potentially affect binding of antibodies, recognizing the canonical RBD sequence. Especially the mutation in E484K in position 484 stands out because an acidic, so negatively charged glutamate has been changed for basic, positively charged lysine. So biochemically, this is a significant change, and this is consistent with the observation that variants containing this mutation, like the Beta, Delta, and more recently also the Omicron variant, reduce the effectiveness of treatment with serum from convalescent patients. Sorry. It is also to be expected that some of these mutations might negatively influence the function of some existing antibodies used by SARS-CoV-2 for SARS-CoV-2 lateral flow assays, which is important in context with rapid tests, or other immunoassays. In order to verify the usability of any antibodies targeting the S protein, it is therefore imperative to test whether they retain the ability to bind the intended target in novel virus lineages. So let's have a closer look at some of these lineages and what the S proteins look like. Shown here from the left to the right are the structures of the S proteins from SARS-CoV-2 variants of concerns alpha, beta, gamma, and delta. For clarity's sake, amino acids that differ from the S protein of the canonical Wuhan wild-type strain are indicated only for the gray monomer in each of the trimers. But I mean, obviously, each of these mutations you would have to reflect to, you would have to project also on the yellow and green subunits shown here. Many of these SARS-CoV-2 variants of concern mutations are located within the S protein's RBD, shown here, just as a reminder. That means the primary interaction interface with the host cell's ACE2 receptor and target for many neutralizing antibodies. So not surprisingly, these mutations have been associated with different ways to evade an antibody-based immune response or improved interaction with ACE2. So the mutations K417N, E484K, and N501, all found here, for example, in the gamma variant, have local destabilizing effects and can negatively impact binding of neutralizing antibodies. That's consistent with the observation that that these variants here have improved escape from neutralizing antibodies. Other mutations might benefit the virus by increasing the propensity for the RBD upstate and facilitate access to the ACE2 receptor. Remember, I had mentioned initially that the RBD has to be in an up position in order to contact ACE2. Alternatively, they can also cause tighter binding to ACE2, as it is the case for the mutations N501Y, E484K, and S477N. And also, to a lesser degree, for Q498R. I say to a lesser degree because Q498R has been shown to only emerge in context of with the N501Y mutation. So the latest addition to this list of SARS-CoV-2 variants of concern is variant B.1.1.529 Omicron, which has accumulated an unusually large number of mutations. In the S protein alone, there are 30 substitutions, 6 deletions, and 3 insertions. So it becomes clear in this representation of the S protein trimer that a large number of these charges are situated again in the RBD. 15 of the amino acid substitutions are concentrated within this only 193 amino acid long domain. As a comparison, the entire protein is 1,273 amino acids long. So before I go further, just a word of explanation. This set of mutations here that I show here is characteristic for the SARS-CoV-2 B.1.1.529 Omicron BA1 variants S protein. So when mentioning the Omicron variant during the remainder of my presentation here, I will be referring only to the BA1 variant, although the Omicron BA2 variant seems to be bound to replace BA1 mid to long term. But at the time when we carried out the subsequently described experiments, no BA2 specific proteins were available yet. So the question remains now here, seeing all these mutations, how do all these mutations affect binding of S protein antibodies regardless of their use in therapy, diagnostics, or research? So a word on neutralizing antibodies. Production of neutralizing antibodies is but one aspect of immunity upon vaccination or infection with SARS-CoV-2. Neutralizing antibodies are also highly relevant for neutralization assays involving pseudotype viruses or virus-like particles. Simply put, a neutralizing antibody gets in the way of the interaction between two binding partners. In case of SARS-CoV-2, the main target for neutralizing antibodies is the S protein's RBD, shown here separately on the right. And accordingly, they get in the way of its interaction with the ACE2 receptor on the host cell surface. Depending on the location of the targeted epitope, neutralizing antibodies are classified in classes 1, 2, or 3. Classes 1 and 2 and class 1 and 2 antibodies block two different epitopes in the vicinity of the ACE2 binding interface. Class 2 antibodies, indicated here in yellow, effectively block the receptor binding motif, RBM. And so this motif consists of amino acids that form direct contacts with residues on ACE2. Here one finds, for example, the residue in position 484 that I had mentioned already repeatedly on the previous slides. And yeah, it has been shown that mutation E484K can effectively abolish binding of class 2 neutralizing antibodies, thus negating the neutralizing effects. So class 1 and 2, interestingly enough, contains a couple of therapeutic antibodies, like, for example, as well this monoclonal antibody from Regeneron. Class 3 neutralizing antibodies, on the other hand, do not block directly ACE2 contact, but instead they are thought to destabilize the S protein trimer. It becomes kind of clear to see here because they're relatively far away from the interaction interface with ACE2. And again, here as well, you one finds some therapeutic antibodies like second monoclonal antibody from Regeneron and the S309 clone. So now finally, the class 4 neutralizing antibodies do not fall in any of the three other classes. They have an epitope which is situated on the flank of the RBD that does not form any contacts with ACE2. Interestingly, the epitope is highly conserved, which makes class 4 antibodies valuable tools for immunoassays. And one of these antibodies is antibody CR3022, about which I'm going to talk a little bit more extensively now. The class 4 neutralizing antibody CR3022 was originally isolated from the serum of a convalescent SARS-CoV patient about 15 years ago. The antibody on its own shows only weak neutralizing effect on SARS-CoV-2, probably because it does not interfere directly with ACE2 binding. It does, however, synergize well with other neutralizing antibodies. That means the combined effect of the two neutralizing antibodies is larger than would be expected. Besides its function as a neutralizing antibody, CR3022 is frequently used as an immunoassay in immunoassays targeting SARS-CoV-2 S proteins, kind of as a gold standard. Shown here on the right is a schematic presentation of the antibodies context, shown here in yellow, of the known epitope within the S proteins RBD, again in orange here. RBD mutations are indicated in the rows above for the different SARS-CoV-2 variants, similar to what I had shown before already. Based on the RBD's primary sequence, the epitopes of antibody CR3022 do not include any of the amino acids mutated in any of the variants, except for the most recent addition of the Omicron variant, which has a mutation in position 371. So unless the quaternary structure of these variants, S protein trimers has an effect on binding of the CR3022 antibody, we hypothesize that the antibody should still be binding to most, if not all, of these variants here. So in order to test this hypothesis, we sourced high-quality, full-length trimeric SARS-CoV-2 proteins, including the membrane proximal stalk from our protein expression lab here in Germany. The cells are expressed in HEK293 cells, so in order to avoid pre-cleavage, the furin cleavage site had been mutated, but other than that, these are unchanged, be it wild type or for the respective variants. The S proteins tested were those of the canonical Wuhan strain, referred to here as the wild type, and the S proteins from the variants alpha, beta, gamma, delta, Omicron, and a couple of variants of interest, so epsilon, eta, and kappa that we had still tested as well. The different S protein variants used and the CR3022 antibody itself are all available through our catalog under the product numbers, which are indicated here on the right. For the measurements, we teamed up with Nanotemple Technologies, and I will show you on the next slides whether our hypothesis regarding binding of different S protein variants by antibody CR3022 held true or not. To quantify the strength of the interaction between CR3022 and the various S protein variants, we used the Nanotemple monolith benchtop instrument, which uses microscale thermophoresis to measure the dissociation constant, or KD, between two molecules. To measure the KD between two molecules with MST, one of the two molecules needs to be fluorescently labeled. Since we had S proteins from 9 variants to test, it made sense to label the antibody CR3022. This way, we could easily test all 9 variants and obtain comparable results. For the MST measurements, we then prepared dilution series of each of the S protein variants in a concentration range between 7.6 picomolar and 250 nanomolar. The diluted S proteins were then mixed with a fluorescently labeled CR3022 at a constant final concentration of 2.5 nanomolar, which is low enough to also allow the measurement of very strong binding affinities. The dissociation constant for each S protein variant to CR3022 was then determined based on the change in fluorescence with the different depending on the different S protein concentrations. So, and here now we see the results of these measurements. Shown are so-called dose response curves with the concentration of the S protein plotted here on the x-axis, and the so-called F norm value, which is the measured signal in MST on the y-axis. Since we want to compare the different S protein variants, it is helpful to look at a normalized binding curve. That means a so-called fraction-bound representation, which looks like this. Normalizing the graphs to depict relative values for the fraction of S protein to the antibody, shown here on the left, makes the differences very clear. The shift to the right, which means towards higher concentrations, so we're going here now from nanomolar to micromolar concentrations, becomes obvious in this representation of the data for many of the SARS-CoV-2 variants. It also reflects the dissociation constants for the antibodies S protein binding, summarized in the table here on the right. Binding of CR3022 to the canonical S protein, shown here in orange, has a KD of around 1 nanomolar. As a reference, an antibody with a KD in the low nanomolar range is generally considered a high affinity antibody. Many commercial antibodies have KDs in the micromolar range. That means about 3 orders of magnitudes higher than this. Some very good antibodies may even show picomolar KD values, typically in the 3-digit picomolar range. So again, that's about 1 order of magnitude lower, so tighter binding. The KD for the B.1.1.529 alpha S protein is virtually unchanged, with about 1.3 nanomolar, whereas the S protein from SARS-CoV-2 variant P1 gamma is already bound with the three times higher KD. The dissociation constant for the B1351 beta S protein with a value of 10.8 nanomolar is 1 order of magnitude higher than it was for the wild type. S proteins from variants B1617 kappa and B.1.429 epsilon have even higher KDs of about 12 and about 20 nanomolar respectively. The antibody CR3022 binds slightly better to the B525 S protein. Actually, sorry, B5 with I didn't even show. I'm really sorry I didn't show this. Anyway, then let me just tell you. So the antibody CR3022 binds even slightly better to the B1525 S protein with the KD of about 2.25 nanomolar than for the P1 gamma variant. We measured the highest KDs for the S proteins of variants B1617 delta and the most recent addition B.1.1.529 Omicron with 51 and 38 nanomolar respectively. Just as a note, this apparent difference might actually not be as big as it seems here, because these were actually done in 2 different experiments, and because this experiment this data I already showed it last year in September, whereas this one is new data. And when doing this, Andreas actually also tested the Delta variant in parallel with the Omicron variant here, and that value was close, and the value for the Delta S protein was closer to what we see here for the Omicron variant. So probably it's not. Long story short, the difference is not as big as it seems here probably. So even though the CR3022 RBD epitopes are not affected by any of the mutations in these variants based on the S protein's primary structure, the antibody's affinity to the tested variants is reduced, maybe due to steric hindrance elsewhere. So that being said, based on these measurements, it's apparent that the CR3022 neutralizing antibody binds still tightly to all tested S proteins. In comparison, the KD for binding of the wild type S protein to its host protein target, so the ACE2 receptor, had also been measured and I'm referring here to a publication using SPR, and it was found to be at about 14.7 nanomolar. So it was a different method. So so I wouldn't directly compare the exact values, but the order of magnitude is the same here. So for CR3022, we had already hypothesized beforehand that the effect of the mutations in the tested S protein variants should be minor. So how does this compare with other antibodies? In order to address this question, we did similar measurements with two other monoclonal antibody anti-S protein antibodies. This first antibody shown here, the clone MM43, has been reported to retain its neutralizing capacity on virus-like particles displaying S proteins from variants alpha, beta, gamma, and delta, and epsilon. That was a Nature Communications paper back in October or September, I think. So based on our measurements, MM43 binds still very tightly to the canonical S protein, again shown in orange here, with a KD in the low triple-digit picomolar range. It all, as I had said, a triple-digit picomolar range is a very good value for when it comes to affinity for an antibody. It also still binds very tightly to the delta variant S protein shown here in blue. In fact, the antibody binds the canonical delta S protein with higher affinities than CR3022 and the delta variant as well. So this is not necessarily surprising, since CR3022 has, as I had mentioned before, a relatively weak neutralizing effect, and this may be in part due to a higher KD in comparison to other S protein antibodies. However, what we can also see for MM43 here is that we could not detect any binding of this antibody clone to the Omicron variant S protein within the concentration range used for our measurements. So I did not forget to represent the pink curve here. We just couldn't measure it. This is a normalized representation of the raw data, and that's why I couldn't. That's why it's just not possible to show the curve here. In the raw data, the pink curve basically stays down here on the x-axis. So to our knowledge, the epitope for this antibody is not known, but it seems to be severely perturbed by the amino acid changes in the Omicron variant. The second antibody from the same source, so clone MM117 shown here, shows decent binding to both the wild type and S proteins shown again here in orange and for the wild type and blue for the delta variant S protein. But the interesting part with this antibody is that it did bind with about twofold higher KD to the Omicron S protein than when compared to the wild type protein here. So the exact values are shown here with about 50 and about 25 nanomolar KDs. It is so I would actually say it might be a better choice for any immunoassays in which reliable interaction with the SARS-CoV-2 Omicron variant is essential. On the other hand, the MM43 antibody might be interesting if you want to differentiate between, if you want to clarify based on an immunoassay if the Omicron variant is present or not, if you have a comparison of assays done with these two antibodies. So here now is a second set of monoclonal antibodies that we tested for the capacity to bind the S proteins of different SARS-CoV-2 variants. Just a quick disclaimer, we have not tested these antibodies yet with the Omicron proteins because these tests were done before we had started with the Omicron S protein. So the first of these clones, SP422, was originally raised against the entire S1 subunit of the SARS-CoV-2 S protein. The exact epitopes that antibody binds to have not been mapped, so I cannot comment on this. As shown here, the antibody binds all 6 tested S proteins with higher affinities. That means lower KDs when compared to CR3022. SARS-CoV-2 wild type and alpha have even KDs again in the triple-digit picomolar range shown here, so very tight binders. The result with the second antibody that we tested, SB185, shown here on the bottom, are however quite interesting. So this antibody seems to bind the wild type S protein and that of the alpha variant even tighter than SP422. So the KDs for both measurements are lower compared to the corresponding SP422 values. However, the KD for the binding of the S protein of the delta variant are already 1 order of magnitude larger. and binding of S proteins from variants beta, gamma, and kappa appears to be completely abolished, and the KDs are not measurable. Again, in the concentration range that we checked. Both clones are raised against, both of these clones are raised against the S1 subunit of the SARS-CoV-2 S proteins, but in case of antibody SB185, it appears that the S protein of variants beta, gamma, and kappa contain amino acid substitutions that are essential for binding of this antibody. So now, as mentioned initially, all these measurements were carried out using MST on Nanotemple's monolith benchtop instrument. Commonly used alternative methods for the purpose of measuring determining antibody affinity to the antigens are surface plasmon resonance, or SPR, or enzyme-linked immunosorbent assays, better known as ELISA. So what are the differences between these three methods, and when should you be using what? So firstly, all three methods follow a titration approach. So that means a dilution series of one of the two binding partners is prepared, and it is observed what concentrations are needed of the other binding partners to achieve binding. The difference here, however, is the readout. MST and SPR are both quantitative. That means one gets an accurate measurement of the KD values between the two molecules, which allows a direct comparison of different antibodies. I mean, that's what I just did over the course of the last slides. I gave you specific KD values that we had determined for the tested combinations of antibodies and S proteins. An ELISA, on the other hand, gets a semi-quantitative readout that only allows a qualitative assessment of the antibody-antigen binding. So you can say at higher concentration it binds better or worse, in which range, but you will not be able to give a specific KD in this case. So next up is the factor of immobilization. MST is an in-solution measurement in which one of the two molecules is fluorescently labeled. In SPR and ELISA, one of the two binding partners is typically immobilized onto a surface. SPR offers different immobilization chemistries, which also allow a site-specific immobilization in certain cases. In the ELISA, on the other hand, the antigen is usually immobilized non-specifically to the plastic. So this immobilization can have unwanted effects, like, for example, denaturation, or just that certain epitopes are covered and the apparent KD is much higher than it actually would be, just because there are fewer molecules available for the actual binding that you want to measure. Then next up is fluidics. MST, I just mentioned it before, is an in-solution method, and it does hence not require any fluidic system. In SPR and ELISA, the solutions above the immobilized protein needs to be exchanged. SPR achieves this with the help of the fluidic system, while ELISA uses multiple wash cycles. Here lies another potential drawback of ELISA, because weak binders can be missed, because if a ligand with a fast off rate dissociates quickly, it can get washed off during initial wash steps and will not be detected afterwards. MST and SPR both do not have this limitation and can even measure weak micromolar binders with ease. However, just to mention, SPR uses a fluidic system, so clogging might be a potential issue here. So it's maintenance intensive. In a significant advantage of MST is the comparatively low consumption of sample. So in our case, the S protein variants are difficult to produce, and one has to be able to work with few micrograms. Difficult to produce means it's very expensive as well. So this becomes even more interesting when testing many antibodies or working with a high concentration to detect even weaker bindings. So having a 5-fold difference here between MST and the two other methods in sample volume can make quite a big difference. So lastly, then we had to comes the factor of time. We had to perform, in our case, we had to perform labeling of each of the tested antibodies only once. The labeled antibodies are then stored in aliquots at about 80 degrees, at minus 80 degrees, not 80 degrees centigrade. And they are then being used without the need to be labeled again. So you have initially you have a time investment of about 1 hour for labeling, but afterwards this you can do without that. So this brings the time per the actual titration then down to about 10 minutes. So in comparison, association and dissociation measurements in SPR take about 1 hour, while ELISA takes considerably longer due to the multiple incubation, washing, surface blocking steps involved. Be this using a robot or doing this by hand, the time is quite a bit longer. So now, how do these measurements done with the different methods compare to each other? So for a direct comparison of MST and ELISA, we had a look at the MST measurements for the SP422 and SP185 antibodies I had just shown you before, and compared those with ELISA data we had also available for these antibodies. So interestingly, the MST measurements gave very comparable results to the ELISA. As you can see here on this slide, both methods show that the SP422 antibody binds the S proteins of variants beta and gamma weaker than that of the wild type and the alpha variants. But altogether in a similar concentration range for both MST and for ELISA. So this is quite remarkable because in MST the antibody was labeled, and we used the S protein in the dilution series, whereas in the ELISA, the S protein was immobilized and the antibody was used as a variable binding partner. So this together is a very strong proof that that what we see here is real and significant. So I mean, having these orthogonal methods for validation is always very useful. Please bear in mind here that the ELISA measurements are semi-quantitative and do not give a clear dissociation constant. Also, the amount of S protein coated on the plates for the assays was considerably higher for ELISA than for the MST measurements. I also would like to mention another advantage of MST compared to SPR and the ELISA, and which is that the measurements can be easily performed under challenging conditions. For example, we, meaning Andreas, also did some successful binding assays at higher temperatures at 39 degrees, and also in the presence of 50% human plasma. So these are the kinds of conditions that can be difficult for other systems, where, for example, the fluidic system can become clogged, as I had mentioned just on the slide before, and where the higher temperature can influence an enzymatic reaction, like in an ELISA. So now, having presented the binding affinity data we measured, how do these insights compare to what has been reported in the real world? That was a bit fast. I'm sorry. So as mentioned before, our primary interest for the experiments was to find out whether antibodies used for immunoassays can still bind S proteins of novel SARS-CoV-2 variants. Not necessarily how does this work now with an immune response upon vaccination or antibody therapeutics. But naturally, this question is also highly relevant for antibodies produced in response to SARS-CoV-2 infection or vaccination, or again for the therapeutic antibodies. So a quick comparison to some real-world data shows that the effect of the S protein mutations is indeed measurable. So in this first study shown here, just what is a note. As I had mentioned initially, this presentation is an update from a presentation in September, and this slide actually stayed unchanged from a presentation I had given then. So there's additional data since then, but this was what was available in September 2021. Okay, that being said, here in this first study from South Africa, the authors came to the conclusion that the variant of concern B.1.1.529 beta does indeed escape neutralization by class 1 and class 2 antibodies. Here in the second publication from Israel, the BioNTech Pfizer mRNA vaccine was shown to efficiently neutralize the SARS-CoV-2 wild type as well as the alpha variant. So in comparison, neutralization was reduced almost by 7-fold for the beta variant. This third article here then from France concludes that the B.1.617.2 Delta variant can escape antibodies targeting epitopes both with the RBD and outside of this domain. So this last series of publications here from the UK looked at the influence of different SARS-CoV-2 variants on the neutralization effect of sera from convalescent patients infected with the Victoria strain, similar to the original Wuhan strain. The authors also checked the effect on post-vaccination sera and sera from patients that had been infected with different SARS-CoV-2 variants of concern. To illustrate the potential influence of different SARS-CoV-2 variants, I am focusing here on one single aspect, which is the neutralization by sera from convalescent patients. While there seems to be no widespread escape of the alpha variant, the sera's neutralizing effects was shown to be 30.3 times reduced already for the beta variant, and fourfold reduced for the Delta variant. The effect on neutralization by different antibodies varies depending on the respective epitopes, similar to what we have seen in our measurements. It is not clear what level of antibody production can be considered as good for response to SARS-CoV-2 infection or vaccination. So a decrease in neutralization as reported here does not necessarily equal a failed immune response. In addition, antibody production is not the immune system's only means to counter an infection. I had mentioned, for example, the NTD of the S protein being highly relevant to mount T-cell response. However, it is notable that depending on the antibodies produced, mutations in the S protein can really have significant effect, significant meaning really measurable in this case. So more recently now, the pressing question is how susceptible is the S protein of the Omicron variant to neutralizing antibodies? The Omicron variant was declared variant of concern by the WHO in the end of November 2021. So therefore, there are not that many systematic studies of its mutations effect on antibody binding yet. So they're kind of just emerging as of now. So one of these just emerging studies comprising work from groups in the UK, Israel, South Africa, Brazil, the US, Taiwan, and Thailand, concludes that antibody neutralization of B.1.1.529 Omicron is substantially reduced or even ineffective for more than, and this was for more than 60 monoclonal antibodies tested. For 10 current antibody therapeutics, the authors find that binding to the Omicron RBD of 4 of them is completely abolished, and for the remaining 6, binding is reduced by the factor 6.4 to 276. So thereby considerably worse. Please bear in mind when I talk about these things that these are all especially with the antibody therapeutics, that I'm talking about monoclonal antibodies. In real life, so in a living body, an immune response will not produce only single antibody clones. So, but based on assays using sera of vaccinated or infected individuals, the authors remarked protection against Omicron. In particular, subsequently to the application of a third of a booster vaccine. So even if binding seems to be reduced for single antibodies, all in all, there is still a reliable immune response. So in context, with the data for the class 4 neutralizing antibody CR3022 that I have just presented, it is noteworthy that binding of a different class 4 antibody, which is clone EY6A, to the Omicron S protein RBD was almost not affected, as was found in this study here. So this reflects our findings with the clone CR3022, and it is also consistent with a high degree of conservation of the class 4 antibody epitope, even with the Omicron variant. So the mutations in the B.1.1.529 Omicron S variant protein seem to affect antibody binding and might therefore compromise the effectiveness of neutralizing antibodies. However, the virion's ability to escape recognition by neutralizing antibodies is only one aspect that might influence the success of the SARS-CoV-2 Omicron variant and its reported higher transmissibility. ACE2 binding through the S protein RBD is a second factor for its success. Of the 15 amino acid substitutions in the Omicron S proteins RBD, 11 form direct contacts with the host's ACE2 receptor. The remaining 4 of these 15 substitutions are all close to the ACE2 to the direct ACE2 contacts, and one can therefore reasonably assume that they might have an effect on ACE2 binding as well to the RBD. So the question is whether binding is more or less tight than for the S protein RBD of the original SARS-CoV-2 strain. So early reports about Omicron suggested that binding might be tighter, and the Omicron RBD has indeed 4 amino acids that are associated with higher affinities, which are the substitutions N501Y, E484K, S477N, and Q98R. Actually, did mention all these already before. These might, however, be offset by other mutations that reduce the affinity, such as the Y505H substitution in context with the Q498R and N501 double mutant. So the authors in this paper didn't really hypothesize beforehand whether binding would be tighter or not, but so they went ahead and measured these, and they came to the conclusion that the Omicron RBD ACE2 binding remains comparable to that of the wild type S protein RBD. So in order to check this ourselves, we conceived of a ternary binding assay in which including 3 binding partners. So the labeled CR3022 antibody was served again as a receptor. The ACE2, recombinant ACE2, was bound to the S protein and is then replaced with increasing concentrations of the antibody. And this change then in fluorescence is then measured, and one can thus indirectly determine also the KD of the S protein of the interaction of the S protein with ACE2. So at this point we have only measured binding of ACE2 to the S proteins from the Delta and the Omicron variant. And also in comparison to the wild type, and that's why this is kind of as a proof of concept. That's why this figure is not as polished as the ones before as well. But according to these early measurements, we can see that binding of the delta S protein to ACE2 is on par with the original canonical S proteins. I mean, they're basically superimposed to the 2 curves shown here. However, binding of the Omicron S protein seems to be considerably stronger, so it's shifted towards lower concentrations in this representation here. So because of the indirect labeling and the strong binding, it's difficult to perfectly quantify the KD for these interactions. But it seems that binding of the Omicron S protein is in the 3-digit picomolar range, so very strong, compared with approximately 10 nanomolar between ACE2 and the wild type S protein, and accordingly also with the Delta variants. So we estimated that the Omicron S protein binds about 20 times stronger to ACE2 than that of the wild type and also of the Delta variant. So at the moment, these results can be regarded as proof of concept, and further experiments are still ongoing. Okay, so in summary, we find that based on our MST measurements, the neutralization antibody CR3022 recognizes the SARS-CoV-2 S protein, as well as wild type S protein, as well as the S proteins from different variants, so namely alpha, beta, gamma, delta, Omicron, and several newer variants of interest. Which means that although the binding affinity of SARS-CoV-2 antibodies to the antigens may be affected by mutations, they can remain valuable tools also for novel virus variants depending on the epitopes they bind to. As we had seen, we had tested different antibodies. The CR3022 bound to all of them, others to some or better, and some better than others, or not at all. So we have also shown that it's essential to verify the antibodies used in immunoassays, that they actually retain the ability to detect the intended antigens in novel virus lineages. We are now working on extending these binding measures to S proteins from other SARS-CoV-2 variants, well, as they come up. And besides, we are testing monoclonal and polyclonal antibodies, as well as different antibody platforms in direct comparison, including synthetic nanobodies. We actually did some measurements of synthetic nanobodies yet, but yeah, I did not show this data at this point now. The little teaser in the end was the ACE2 binding assays, and these assays are actually still ongoing, and we expect to have more exciting data from this in the future. So as the measurements with the various antibodies show, it's important to have access to a wide range of antibodies to assure reliable detection of the various SARS-CoV-2 variants, and also to be able to tell them apart, maybe even based on immunoassays and not solely known on other methods like sequencing. So therefore, we at Antibodies Online are constantly extending our offering of SARS-CoV-2 related antibodies, proteins, and assay kits also. And while this presentation was exclusively focused now on the SARS-CoV-2 S protein, we are obviously also offering a plethora of products related to other SARS-CoV-2 proteins. This concludes our webinar today. I thank you very much for your attention. In case of any questions or interest in any of the products mentioned here or beyond, please don't hesitate to get in touch with either of us with the indicated emails here. And I am certainly happy to address any questions you might have now.